All right. So today you're going to be, as I've told you before, and as you've done the pre-lab, you're going to be working with a diffusion lab. And in that diffusion lab, what you're looking at, there are two major factors. Wait, hold on. So there's two major factors that you're dealing with. There's surface area and there's volume. Two major factors you're dealing with in this lab, just like you did in your pre-lab. Now you're gonna I've made an agarose gel. I'm gonna cut out cubes. They're not gonna be perfect cubes. Some of them are gonna be elongated. Some of them will be close to perfect cubes. And they're not selectively permeable. They're just going to allow things to go in from high to low. In other words, they're going to be doing diffusion. They're going to allow diffusion to occur. Okay? They're going to allow diffusion to occur. You're going to put these cubes in a beaker and you're going to see diffusion occur, and you're gonna calculate the rate of diffusion. You're gonna try to figure out what is or is not going on. All right, so when you're looking at diffusion, if you have this thing and you want to know how fast it diffuses, remember, and why is this important? Why is it important to know how quickly things diffuse? No, but it's not too far off actually, but not more directly. So you got these things called cells, correct? Yeah. And they're generally round, but they can be cuboidal. They can look something like this, all right? They have different shapes depending on what the tissue is, right? The lung tissue or the skin tissue or the eye tissue. The cells can take on different shapes. For instance, a neuron can look something like this. Well, that's a horrible drawing, so let me redraw that. A neuron could be very long and have an axon and a dendrite, and it can have a synapse. And it still has all the same. All the cells in your body still have a nucleus, an endoplasmic reticulum, rough and small, Golgi body. They still have ribosomes. They still have membrane-bound organelles, et cetera, et cetera. But some of them have different shapes. But all of them have to get oxygen, right? All the cells need oxygen and other molecules from the outside and go all the way into the cell. How does that happen? That happens through what? It transports, that's right. And through what method is oxygen going to go into the cell? Diffusion, that's right. So diffusion. Because oxygen is nonpolar and small, it does diffusion, just like in the pre-lab, just like in the practice questions, just like in the project that we did all week. Small and nonpolar go right through the membrane, no problem. So no matter what cell, so what do you think? So it's important to know if you have these cells that need these nutrients, that need these things like oxygen that goes through the membrane, goes through every part of the membrane. There's the factors that impact how quickly oxygen and other molecules get into the cell. It's important to know that, right? It's important to know that. So that's why this lab, that's how it applies to biology. The problem is we can't see oxygen. So we need something. So we're going to look at something else. We're going to look at hydrogen ions. Hydrogen ions. It's a hydrogen atom without an electron. It's specifically a hydrogen ion is the same as a proton, just a proton. Now, when you're dealing with water, H2O, naturally in dynamic equilibrium, right? We talked about this a lot this first quarter. Dynamic equilibrium, you have water going from water, H2O, to OH negative and H plus. That's normal. That's water breaking up. Now, it goes back and forth. So you have about 50-50. Even when it's broken up, you have about 50% hydroxide and 50% hydrogen. So when everything's equal, when they're about equal amounts of OH and H plus, we call that neutral. If you have a lot of hydrogen ions, of course, here you've heard of this, I'm assuming, this term pH. Yes? Okay. So you have there's this neutral where the amount of hydrogen, the concentration of hydroxide ion and the concentration of hydrogen ion are equal, right? Then we call it neutral, where H plus is equal, H plus concentration, the square brackets mean concentration, is equal to OH negative concentration. All right, we call it neutral. When the H plus concentration, hydrogen ion concentration, is high, and I'm going to do that with an up arrow. Is that okay? When it's high, we say it has a low pH. Low pH. So this whole region from neutral, which is 7, all the way to 0. So from neutral to 0, we call it an acid. Does that make sense?
What did you call it when it was going up again?
We're gonna see it in a minute. I haven't written it yet. Does that make sense to everybody? So 7 is neutral. 7 is neutral. And OH negative is low, but hydrogen ions are high. So OH negative is low. This is only one definition of the acids. The upper class and the AP kids know what the others are, but you don't have to worry about that right now. All you got to worry about is this idea that when you have a low OH and a high H plus, we call that an acid. Okay? So that means a pH is equal to a pH of less than 7. It's an acid. Now, the opposite is true in the other direction. So in the other direction, when all the way up to number 14, anywhere from 7 to 14, we have a low hydrogen ion concentration and a high OH concentration, OH negative concentration, hydroxide, one of those functional groups I told you had no, OH negative, hydroxide, high hydroxide concentration, low hydrogen ion concentration, we call that a pH. The pH then, pH is greater than 7, so any pH greater than 7, and we call that a base. So it's basic. So we call that a base. Is everybody okay with that idea so far? Seems pretty straightforward. So if you had a beaker and if you had a bunch, how many green dots would you have a beaker that was acidic? You'd have a lot of green dots, which represent hydrogen ions. And you'd only have just a few of the red dots, which are hydroxides, obviously in water. On the other end, in the base, on the base side, if there was a beaker of water, then you'd have the opposite. You'd have a lot of hydroxide ions and just a few of the hydrogen ions. So that's what we're talking about when we're talking about. But here's the problem, is that we can't see OH negative. And we can't see hydrogen, right? Because these are atoms. We don't see these. So how do we measure it? How do we see it? So we need something to show us. We need something to... have you ever heard the word indicate?
Yeah.
What does indicate mean? Yeah,
Something that tells. Gives a sign or something. Yeah.
So what?
Gives a sign.
Gives a sign, right? So when your mom's turning left, what does she do in the car? The signal, or another word for that, by the way, is an indicator. In fact, when you read the Ohio State law, that when you're going to your driving classes and taking your driving test, they don't call it left turn signal. They'll call it left turn signal. And in other places, they'll call it the left indicator, because it indicates which way you're going. Correct? All right. So we can't see this, so we need something to show us. So we have this indicator that's called, and this is just one example. There's other, you've heard of pH paper, right? Who said that? Who's that? All right, next time, you raise your hand. You don't just jump in, and I'm going to leave it. I'm recording it now so everybody knows you just jumped in and say it's time to go like that. That was really rude. But I'm going to let it go. We're good. So it's time for lunch, is that right? So go ahead and take your apron and your goggles off, go to lunch, come back, and we'll finish this. So, for example, we can use this indicator called phenolphthalein. And phenolphthalein is a molecule, and I'm going to draw it as just this kind of pink dot. It's a molecule. Oops. It's a molecule that changes color when in different conditions. So if this molecule is in a basic solution, if it's in a basic solution, and remember that means a pH that is greater than 7, so there's not a lot of hydrogen ions, then this molecule is pink. If this molecule is in an acidic, neutral to acidic, then the pH, where the pH is less than 7, then it's clear. So it has no color. So this molecule can actually absorb all the light except pink when the pH is greater than 7. So the other thing you have to know is there's this thing called ammonia. So ammonia is NH3. So that's a nitrogen with 3 hydrogens.
What is it? Filter form.
So ammonia, what's interesting about ammonia is that it's called a base. So what it does is it takes the H pluses out of the solution, leaving the OH negatives in there. And so that's what we call a base. In ammonia, phenolphthalein is going to be pink. So step 1, we have an indicator that is pink when it's basic in the solution. Step 2, we have a basic solute, NH3. It's a molecule that's basic. So what we can do is, and what I did was I made a gel out of agarose, and agarose is a seaweed protein. Agarose is a seaweed protein. Acts a lot like Jello gelatin, and I made this. And remember what we talked about when we did paper chromatography, etc. We have these long fibers. Is these polymers of amino acids that have created a network that's going to allow us, and ultimately you can form a whole cube out of it. And I poured it into a mold, right? So that's what it would look like if we could see the molecules of protein. It would look something like this if I did a cube of it. So what I did is I poured it into a mold, and then in that liquid, before I poured it, I had this big flask. Let me draw this correctly there. And in that flask, I had the agarose as a liquid before I poured it, and I added to it phenolphthalein. I added ammonia, and I added agarose to water, the three together, and then I let it cool in the refrigerator, and what that gave me was this pink gel, just like Jello gelatin. So as long as the gel is basic, as long as it's pink, it'll be pink. So we got this big slab of agarose gel that's kind of solidified, like Jello that you eat at home. Some of you've eaten at home, and it's pink, so that's great. Now I'm gonna cut out cubes, and there's gonna be 3 cubes, and it's going to be a big cube. There's going to be a medium-sized cube. And then there's going to be a small cube. And of course, all 3 of them are going to be pink because that's how I made them. I put phenolphthalein in there with ammonia, so that I did that on purpose to make the gel pink. So I know that it's basic. How do I know it's basic? Because it's pink. If it turns neutral or acidic, it's going to become clear. So the only way this is going to change color, is if it comes into contact, if it becomes acidic, if you start to add hydrogen ions. So we have to add hydrogen ions if you want this to change to clear. So one other thing you should know is when you add an acid, when you add an acid to a base in a chemical reaction, when you add an acid to a base, what you make is salt and water. This is called a neutralization reaction. So in a neutralization reaction, what happens is that you add a base. Anytime you have an acid, if you add a base to it, it'll release a little heat, and what'll happen is that the H's, the OH and the H pluses are going to come together, and they're going to make what? OH negative plus H plus makes what? Water. So you're going to make water. So when you add an acid and a base, you make water, and then the other parts become the salt. Whatever, and it all depends on which acid and which base you're making. So what we're going to do is we're going to add hydrochloric acid, which is HCl. Hydrochloric acid, Cl, lowercase l. Remember when you're talking about the periodic table, if there's a capital letter, that's an element. If there's a lowercase letter, that's the other part of that same element's name. So hydrogen only has an H, but chloride has a Cl. So hydrochloric acid is HCl. And I'm going to add it to ammonia with NH3. And then when the ammonia meets the hydrochloric acid, you make NH. Didn't record. I don't know why that didn't record, but it's hard to guess this one. But what happens is that the H plus leaves, because hydrochloric acid is what we call a strong acid. And it releases the hydrogen ion, gets released, and it forms, and when it mixes with ammonia, it makes NH4 plus and then Cl minus. And those two kind of stick together. They form ammonium chloride. That is a salt. So that's the salt. Of course, this is happening in water. Okay. In another reaction, if there was to give you a simpler version of this that would help you understand what a neutralization reaction is you can say HCl, same acid, and mix it instead with sodium hydroxide. Now, this is the simple version of it. What would happen is you'd get, what would you make if this H plus comes out and then the OH negative comes out? What's left? NaCl, you call it table salt, the stuff you sprinkle on your food, plus what? OH negative and H plus make water, H2O. So those two together, then what you get is salt and water. Is that clear to everybody? So that's normally what happens, but of course with ammonium chloride, something else is going on. You get kind of an association. So the H plus mixes with the Cl minus, and they form ammonium chloride. It's a salt. They kind of stick together just like the NaCl does. All right. So that's the idea. But when that happens, they neutralize each other. So this is actually, since this is a plus and a minus equals plus and minus 1 minus 1 equals 0. So this is neutral, all right? It's not impacting acidity. You don't have an H plus floating around anymore. As soon as I added the hydrochloric acid, HCl, as soon as I added HCl to the water, it broke up. I have a lot of H plus ions in there. So when I have this beaker, when you have a beaker, you have an acid. You have a strong acid, so you have to be very careful. Is that clear? So there's H pluses floating around in here, all kinds of H pluses, and then there's some Cl minuses floating around in here. Okay, and of course there's water, right? H2O. And we could talk about other things, but we're not going to talk about all that. That's chemistry. But just the water is the solvent, right? The solute's the HCl. So a lot of hydrogen ions. So this is an acid. It's a low pH. So a pH lower than 7. All right? Okay. So I take my cube. Let's say I take a small one. And I put it in there. I take my cube and I put it in the solution. The solvent water, H2O, is out here. It's blue, okay? Then I have the Cl, the chlorides out here. A bunch of chlorides, right? Negatives. And I have then also I have H pluses, right? What's going to happen? What's going to happen when I put the HCl, this cube, that is basic, right? That has ammonia in it, right? You remember that? It has phenolphthalein and ammonia, correct? Yes or no?
Yes.
Okay, what's going to happen when I have all these ions, this H pluses and negative and Cl negatives out here? What's going to happen? You tell me. I'm waiting.
Is it going to neutralize?
They will, but how are they going to neutralize? How are they going to get to the base? Where's the base? It's in the gel.
So it's going to go through the gel.
It's going to go into the gel. Why? Why would the H pluses go into the gel? Hint, you already know the answer. How would the H plus get into the gel? Do you remember the gorilla in the movie? I showed you the gorilla in the movie, didn't I? All right. You didn't show you the gorilla in the movie. Record. We know they're gonna neutralize each other. They're outside. We know that this is a gel, just like paper chromatography. What is happening in this lab? What's going to happen with all these ions in the solution out here? Where you said they're going to neutralize the base. You're correct. But how is the H plus going to get to the base that's in the gel? It's permeable, so what's going to happen? Why? Why would the hydrogen ions go inside? What's making it happen? What's the force? Why does anything move?
Because the kinetic.
Diffusion, because of kinetic energy, you're correct. Because of the process called what? Diffusion, for God's sakes. Diffusion. You guys keep watching a basketball and missing the gorilla. I'm serious. That's a real problem. The big point here is diffusion. All this other stuff is how we see diffusion. How do we see diffusion? Well, you're about to see it. How do you see it? You have to understand the idea that as the hydrogen ions go into the gel, what's going to happen? As the hydrogen ions go into the gel, what's going to happen? It's going to neutralize the base, and then what happens?
It reaches equilibrium.
Well, that's correct. But what happens when the gel, when the ammonia is neutralized? The gel's going to change color from pink to what? To clear. Now you see the whole picture, don't you? Now you're counting the basketballs and you're seeing the gorilla. You see both. You can't focus on one part of the problem or you're not going to see the whole picture. That's the point. So you're going to plop them in. Now what's going to determine how fast it happens? How fast diffusion happens? Two things. The surface area, which is this, how big this is, and the volume. That's how fast this cube is going to change shape. The two things together. How do you find the surface area of a cube? Well, let's go back to our original drawings. We got two cubes, so we got two problems. So if we had a perfect cube, if it's a square, that's easy, right? That's easy. If it's a 2 centimeter by 2 centimeter by 2 centimeter, that's a pure cube. And it's a perfect cube, no problem. Then the surface area is easy, right? I got each side. What's the area of each side of a cube, of this cube? What's the area? How do you calculate area? Length times width. So length is 2 centimeters, width is 2 centimeters. So 2 times 2, that's 4. But that's only of one side. One side. How many sides? And you did this already in your pre-lab. Or maybe you didn't because you didn't do it. You just copied. Okay. But if you did it, you know that if you have one side is 2 times 2, that's 4, right? Centimeters squared. How many sides do you have in a cube? 6. So you multiply it times 6, which 4 times 6 is what? Is what? 24 is correct. So you have 24 centimeters squared. That's the surface area of this cube. And volume's easy. What's volume? Length times width times height. So 2 centimeters times 2 centimeters times 2 centimeters. So that's equal to 6, geez. To 8, that's 2 times 2 is 4, times 2 is 8. 8 centimeters cubed. So now you have 8 centimeters cubed. You have 24 centimeters squared. You have surface area. And you have volume. Now what you can do is calculate. Now all you got to do is calculate surface area to volume, which is 24 divided by 8, which is what? 3. So the answer to surface area divided by volume ratio for the big in this example is 3. That's easy, correct?
Yeah.
The problem is that some of your cubes won't be perfect squares. So some of your cubes will be longer than they are wide. So now, how do you do that? Well, it's still the same idea, guys. It's the same idea. You see the drawings are up on the board already for the other examples that we had done before. But that would mean you have 4 centimeters here. This is 2 centimeters. And this is 2 centimeters. How do we find the surface area here? Well, we do the same thing. What is area? How do you do area of a square? Or rectangle in this case? Length times width. So surface area of the long side, right? Surface area of the long side is equal to 2 centimeters times 4 centimeters, correct? So that's 8 centimeters squared. How many long sides do I have?
4.
4, correct. I have one here. I'm going to label it A. This is the long side. Now there's another long side over there. And this is the 4th long side here on the bottom. Correct? So 4 sides. So 8 times 4, because there's 4 sides that have that length, is how is what? 32 centimeters squared. Is that all the surface area on this cube? No, that's only 4 of them. I need the other 2 sides. And those 2 sides are over here. There's one. The other one is over here. So this is side B. Side B, there's 2 of them. Their length, they're 2 by 2. So 2 by 2. So the surface area of the short sides is equal to 2 centimeters times 2 centimeters, which equals 4 centimeters squared. Now what do I do with those to get the total surface area? I add them. So that equals 32 and 4 is what? 36 centimeters squared. That's my surface area. Now my volume is still easy. It's as easy as it was with a perfect cube, correct? My volume is what? Length times width times height. So 4 times 2 times 2. So 4 times 2 is 8. And 2 times 4 is 8, times 2 is 16. So 16 centimeters cubed. Easy peasy. Lemon squeezy. So surface area is 36 centimeters squared. The volume is 16. So now all I got to do is take 36 centimeters squared divided by 16 centimeters cubed. And that is my ratio. And actually in a ratio, there is no real units. You don't have to worry about this. So 36 divided by 16 is, somebody pull out a calculator, tell me. 36 divided by 16? Nobody? I'll do it myself. You guys are too slow. How is someone so young move so slowly? I do not know. 2.25 is the ratio of surface area to volume for this one, where it was 3 for the small one. Isn't that interesting? The smaller the cube, the bigger the surface area to volume ratio. What that means is that when you put that cube in this acid, the smaller the cube, you're still going to get, the surface area tells you how fast they can start to go in on all sides, but how much they have to actually neutralize depends on how fast they can get into the center, depends on how much volume there is on the inside. So the larger your volume inside compared to the surface area is going to determine how quickly this cube gets neutralized. So we're going to make 3 cubes real quick. Hopefully we can at least start to see it, even if you can't measure time. You're going to have your chart that you've already filled out. I just showed you how to do the calculations. You're going to measure time. As soon as you put the cubes in, you start a stopwatch, you start to measure time. As soon as it changes color completely, you stop the stopwatch. But since you're using one stopwatch for all 3, so once the cube becomes clear, you write down that time. When the next one becomes clear, you write down that time. When the last one becomes clear, you write that down that time. So you have a small, medium, and large. That's it. And then your lab write-up is explaining what you saw in your own words. All right, I'm going to go on. Let's start the lab.